So we're going to lyse the cells. It can be through osmotic osmosis, right? Just water-based, so you can break open the cell. And then you can have even equipment, mechanical disruption. There's plenty of different types of instruments that can break open the cell. So all of these are pretty much breaking open ways to break open a cell. Once you have those cells broken, then you want to centrifuge it. Centrifuge is spinning. So this is the case where you have the cells all in a homogeneity. Everything is in here in the soup. Then you're going to have to centrifuge it. When you centrifuge it, you will see that you go from a slow speed to a high speed. So when you look at the first pellet, the reason you're centrifuging is try to figure out where this protein is localized. Is it a nuclear protein? Is it a protein found in the mitochondria? Is it a protein that's found in the lysosome, the smaller microvessels? Right? So you want to try to figure this out because you're not going to take this entire pool and fish through it. So you want to make your life as simple as possible. So you go from slow to all the way to high speed, and with the slow speed, you know that the nucleus is the largest organelle, so it's going to come down fastest. So you don't need to have a high speed. That's why it's at about 500 G. Next, you separate out, you get mitochondria, and you know that the microsomal fraction, like the lysosomes, will be the smallest and they will plummet down the last. So that's why you see here you have to have a higher speed to get that. So each time you get that, you have a pellet. If that's the pellet of your interest, you just take out the rest of the homogeneous soup here, and you just take the pellet that you're looking at and then go for further isolation. Okay, so it all depends on where your particular protein is going to be found within the organelle. So once I'm going kind of backwards, I'm pretending my protein has been purified, or at least I have something. I'm just not going to take the protein and pretend that it works. I have to test it, right? So this test is called an assay. So that's going to tell you, yes, I have some protein present, but is it active? Because you've gone through so many processes of purifying, you may have denatured it. If you didn't put it in the right buffer, maybe you denatured it. Maybe you didn't purify the right protein. Okay, so you do need to figure that out, and that's called an assay. So when we look at an assay, this book gives you a nice example with lactate dehydrogenase, and there's actually an enzyme that you're going to learn next semester. But it takes lactate, oxidizes it into pyruvate. And what's released is this coenzyme called NADH. So what we detect in this assay is not how much product is made, but we're going to detect whether NADH is being released. And how that is done is through a spectrophotometer. So it's an instrument to detect through light-based how much NADH is being released. So if NADH is being released, it tells you that lactate dehydrogenase is functional. You get that information, that it works. Right? So whatever you purified is functional. So you're really not saying how much pyruvate you're making. You are saying that this is functional based on the release of NADH. Does that make sense? Okay, you're not looking at product formation at this point. So that's an assay. That's an example of an assay. There are obviously different types of assays. This is light-based, using a spectrophotometer. But obviously there's different types of assays that you can do to figure out if your enzyme is functional. One of the most important things is looking at specific activity. So specific activity is the ratio of enzyme activity to protein purification. I'm giving you this definition right now, but I'm going to go through the process of all the techniques you need to know, and at the end I'll show you a table. When you purify a protein in the beginning, you'll have a soup of lots of different proteins, and you're going to be fishing to figure out where your protein of interest is. So in the beginning, your activity will be quite high because it is the ratio of the enzyme activity over your protein concentration. So the concentration of that protein is going to be high when you initially start. Once you go through that protein steps, It could be 2 steps, it could be up to 5 steps, or it could be more. Once you go through all those steps, the purified protein that you get should be your protein. And you won't have all the proteins in the mix. Alright, so the specific activity at that time will increase because yes, you will have a smaller amount of protein, which would be your protein, and your concentration will be lower. So put a lower number in the denominator of this ratio, the specific activity begins to rise. Is that good? Okay, I'll show you that example. I'll show you with numbers when we get to it. Okay, so you ready for the methods that you need to know? Solubility, all right? So that's based on really the salt conditions that I'm going to look at. This is actually a crude method. It's not the ideal method because you can break your protein and denature it. Size, we have a technique that is going to be used to separate by size. Charge, we're going to be able to separate proteins by charge. And binding properties, that's called affinity. So we have methods to purify proteins. As long as we know what that protein binds to, we can use the last method as well. And of course, you need to know something, a little bit something about the protein. So like I said, the salting method is kind of crude. You can denature your protein, but you might want to use it just to filter things out and get some, even if it's false positive, get an extract out. And so that's based on the solubility of the protein for a particular salt. So the solubility of the protein of interest is independent of the solubility of another protein. So each protein will have its own salt concentration where it can precipitate out. Okay, so when you look at this graph here, y-axis is protein solubility, x-axis is salt concentration. So in the beginning, the protein is salted in, which means it's still soluble. As you increase the concentration of the salt will be competing with the protein for the water, and it'll precipitate out. All right, so it'll start to precipitate out. And that's what you're seeing at the end. So when it begins to precipitate out, there's going to be some salt also present within the protein soup. So you want to make sure you get rid of the salt, because you don't want that salt to denature your protein. So you use a method called dialysis, and you take your dialysis bag that has your protein, and you put it into water, and all these salt concentrations, that are in blue, will go out. And so hopefully you have enough of the salt that's out, and you have not contaminated too much of your protein. So like I said, this is a crude method. There's a possibility you can denature your protein, but if you do a good job, most of the salt will come out through the dialysis method. The next, let me know if I'm going too fast, okay? The next method is called gel filtration. So gel filtration is based on size. Okay, so do you guys do chromatography here? You do? Okay. All right. So you've got a chromatography filtration here, and you throw in your protein sample, so it's a little bit of a pool of proteins, and you let it go through, and you're going to filter it out, okay, into test tubes. The principle is size. So when you look at this diagram here, the large ones, the large proteins, will flow through fastest, and the little proteins, the ones that are smaller molecular weight, will get trapped in the beads. And so you do need to know a little bit about the protein. So in these collections, removing the large proteins will come out. So obviously you need to know something about the molecular weight of the protein. And then once you know the molecular weight of the proteins, you can actually collect the test tubes, because if it's a large protein, it's going to come off first. So you can target those specific test tubes and then fish out your protein of interest with another method. Okay, so the large ones come off first, the little ones get trapped, and then you can actually get those little ones out if you need to from the beads. Okay, so that's size. Charge is ion exchange. Okay, so with the ion exchange, you have a column, and the beads can either be negatively charged or positively charged. In this particular case, you have a bead that's in gray, that's all negative charges. So what does that tell you? When you throw in a pool of protein, and of course, you have a buffer within it at a specific pH. So proteins that have, at that particular pH, so this goes back to your PI. So protein thrown into this buffer that has a positive charge, for instance, +2, will bind tightly. Then a protein that has a negative charge. So this protein has a -2 charge, so it'll flow through fastest. This protein here will get stuck to the beads. Okay, so this is all based on charge. You can also have a bead that is positively charged. And if it's a positively charged bead, then you know that the negative charge on a protein will get stuck to the bead and the positive charge will come off. Okay, so you can have either of those conditions. Okay, so it's the same principle, chromatography. You have different sets of types of proteins, and through fractions, you collect them, and as long as you know a little bit about something of the charge of that protein at a particular pH, you'll at least have an idea of which test tubes to look at and isolate it even further. What I need you to know is cation exchange media, not specifically the names. I don't need you to know the names. These are beads. Okay, so these cation exchange media have negatively charged beads. All right, so you need to know that cation exchange media has negatively charged beads. That's opposite of anion exchange media, and that's going to be positively charged beads. Okay, so those two terms you need to know. Okay, I'll come back to why you need to know that, because that's going to come back to your amino acids. Okay, so here you have your ion exchange. Like I said, all the charges here are cation exchange, negatively charged beads, right? So this would be an example of a cation exchange, because the beads are negatively charged. The positively charged proteins will bind to the beads, negative charge. This one will come off first. It has a negative charge, right? It will come off first. Charge of a -2. And then the next one will come off. And so forth. All the positive charge will get stuck on the beads. So how do you get the ones that are stuck on the beads? You add sodium, which is positive. So those are in red. So positively charged sodium will compete for the beads that are bound. And you can actually elute or remove the protein that's positively charged that way. Okay, so it's a competition method to be able to remove anything that's already bound to the beads. Does that make sense? Are you ready for amino acids and how to apply it? Or did you need me to go back to the ion exchange? Everything good on ion exchange? How that? Okay. All right. Let's look at a example, and I'm telling you that this is a cation exchange column. So what does that tell you? It tells you that the column is negatively charged, right? The beads are all negatively charged. I told you when you look at the PI, when you're doing a PI problem, start with a pH of 1, unless I tell you. So I'm telling you in this case that it's starting at a pH of 7. So you go back to what you know about PI and how to solve the problem for PI and be able to use it for this question of what is the order of the amino acid that'll come off from the column, first to last. So the 3 amino acids that have been put into this column are aspartic acid, serine, and lysine, which means you need to know a little bit about the structure, the R chain. And I've given you the pKa value. So aspartic acid, when you draw it out, this is the structure at a pH of 1. So everything is positive, right? Everything has a proton to it. So here's a hydrogen carboxylic acid, and this amino group is positively charged. Okay, so that's what you know. Then you're going to remove a proton at a time, like you've done before. So here, now aspartic acid, so if you go back to aspartic acid, it's 4.1. This is about 3.1 is this table. And then amino group is 9 or 8. Alright, so I'm going to remove it one at a time from a pH of 1. So when I remove it, and I'm telling you, go up to pH of 7. So at this point, anything below a pH of 7 comes off. Right? Which means the hydrogen from this carboxylic acid end will come off. The hydrogen from this carboxylic acid will come off. I don't do anything to this one because it's an 8. Right? I go up to a pH of 7. So because 3.1 and 4.1, 3.1 and 4.1 are below pH of 7, those hydrogens will come off. So here they've come off, and I've got a negative 1 charge. I do the same thing with serine. Okay, remember the serine structure has an OH group to it. I didn't draw this out, but it's not part of this list of has an R group. But all serine will have is the amino group and the carboxylic acid group. All right, so remember the structure of serine. I'm just going to draw the. Okay, so this one does not come off, right? Because it's not part of the 7 amino acids that have as an additional. But the only thing that you're going to be dealing with is the amino group here and the carboxylic acid. Okay, so when you do that, nothing will happen to the amino group because it's positively charged and it's a pKa of 8. So this one is the only one that comes off, right? So this one will come off and it'll be negative. And so you have a neutral charge of 0. Lysine, when you look at lysine, here's the structure for lysine. You go up to, let's see, this one's 8. And this is lysine is here, it's 10.8. Okay, so the only thing that'll come off is which one? At pH of 7. Which one is the only one that'll come off? Just tell me which one will come off. 3.1. Okay, so this one will come off. So there it is. So that one is left at a +1 charge at a pH of 7. So when you go to that, and you go back to what I asked you, the cation exchange column, which is all you need to know is I'm going to tell you it's positive or negative. When you see the word cation exchange, you need to know it's negative. Okay, so some students will not know this, they'll raise their hand, I just can't answer that question. Okay, so you need to know that. So that's your negative charge. So when you look at these 3 amino acids, aspartic acid, serine, and lysine, the one that'll come off first is aspartic acid. Good? The next one that'll come off is going to be serine. There's serine. Because it's neutral. The one that's going to get stuck to the bead is your positively charged lysine. Okay, so there's your lysine. Okay, so how do you compete off the lysine? It's already stuck to the beads. All you have to do is add something that's positively charged ion, like sodium. And it'll come off. Is that good? Okay. It's not enough just to know how to do a PI problem. Alright, that's what I was telling you about. You need to understand what's happening at a different pH. One of the ways to do that is actually knowing the structure and being able to pull out the protons based on the fact that I give you a table, you'll be able to pull out the protons. Do you guys have any questions about this? Okay. If you do, and you look through it and you go home, if you have questions about it, I'll go through it again during review, okay? So just let me know. Okay, so that's ion exchange, affinity chromatography. Everybody heard the term affinity before? A liking for, something that likes something else. So in this particular case, you need to know if the protein binds to something else that it likes. Okay? So in this case, this protein here in yellow likes to bind to glucose. That's why the G is there, that's glucose. So what you do in that case is you create a column with a bead that says glucose stuck to it. And then you throw in your pool of sample of protein, and anything the protein that likes glucose will get stuck to it. You may have multiple proteins, you never know, right? So it gets stuck to it. So that's why we do plenty of different types of methods to purify proteins. So once those proteins are stuck, because it has an affinity for glucose, then you have to release it, right? A competition again. You can add more of the same. You can add more of the glucose molecules, right? So you can add more of the glucose, and the protein from here will bind to the free-floating glucose and will come off. That's how you complete it off. So that's affinity. Okay, so that's one method to compute it off. So this is basically the same thing. You have a pool of samples, you put it into a column, and you figure out which is your protein of interest. And of course, this particular protein will bind to something S. So the S is bound to the beads, and you throw in your pool of protein. So P1 is the protein that likes to bind to S. That's what you're trying to purify. So the rest of the protein, P2, P3, will come off. P1 will bind to the protein that has the S to it, the beads. Once you do that, then you have to, obviously, it's stuck to the beads, so you have to remove it. Like I said, you can add more of the substrate, or the S, to remove it as a competition. You can add it if you know that it binds to another ligand, something else as well. You could throw in that and remove it from the beads. Or you can alter the pH, the usual things that you're aware of, ionic strength, or even temperature. Okay, so that's just a way to compute it off. Do you guys have any questions about any of those four methods? Okay, so if you want to look at a similar problem for a cation and anion exchange, it's in the sample exam, try to do those. Okay, so this, between this and the other problems, the other problem you should hopefully have a good handle on how to work on some of those types of problems and apply it. All right, so if you have no questions about those four, I'm going to move on to different ways to purify protein based on techniques that use antibodies. All right, so the methods that you need to know are ELISA. So I did put several videos on ELISA for you. Application-wise, it's used in pregnancy tests. You can use it for HIV and COVID. Okay, so there are actually different models and uses of ELISA. And the other one we need to know is Western blotting. And for each one of them, of course, you'd have to do the enzymatic assay. I use hormones as an example for that to measure biological activity. So let's go through electrophoresis first. You guys going on to the biochem lab will do gel electrophoresis, and actually you won't be working with proteins next semester, you'll just be working with DNA, but the principle is the same. So in an electric field, a charged molecule will move with a velocity that's dependent on the charge on the molecule and inversely related to the size. And pH is going to be very important as a determining factor for the net charge again. So you're going to have to make sure that this particular gel is in the appropriate buffer. So these are the different types of media. The first one, polyacrylamide, is for protein separation, and you guys will be doing agarose, which is for DNA or nucleic acid separation. So the way this technique works is bigger molecular weight, whether it's protein or DNA, will move slowly. The smaller ones, or compact molecules, will move the fastest. Okay? So that's what I'm showing you here. So here's what the gel will look like. This is a protein gel. Your gel, if you do agarose, will be horizontal. So this is your protein. You load the protein in these are wells. below the protein in the socket here, and the proteins will run through an electric current. So what you're seeing is the bigger molecular weight protein will be on the top. Smaller molecular weight proteins will be on the bottom, and this is your middle size molecular weight proteins. Good? The principle good so far? Okay, so just giving you an example what you guys might see. This is all protein, but this principle also applies to DNA as well. So again, you may see different sizes of the protein, right? And how are you going to figure out the size of the protein with all these bands? These are all bands. How are you going to figure out the size of the protein? I'll come back to this negative and positive charge in a minute, because they are going to be moving based on charge. So that gets to me to the charge. So these buffers will contain what's called sodium disulfide, sodium dosyl sulfate, which is a detergent. And when you look at this, this is a structure. What does this remind you of? You have an alkyl group, and you have a charged group. What does this remind you of? Yes, it does. What's the term for those types of fatty acids that have both a polar and non-polar? Amphipathic. You guys told me in your assignment. Okay, so there's another example of an amphipathic molecule. Don't ever forget the chemistry part of the biochemistry. Okay? A lot of students do forget there is a chemistry component to it. Okay, so another thing we do add that you know of, we've talked about it several times, is beta-mercaptoethanol. And so what that does is, remember a chain of a peptide can be 2 polypeptide chains, 3 polypeptide chains, right? So last time we talked about hemoglobin. That probably would not be the best example. Let me talk about insulin receptor, because insulin receptor has disulfide bonds attached to it. So insulin receptor has disulfide bonds attached to 2 polypeptide chains. But how would you know? Unless you separate those 2 chains out. And you know that beta-mercaptoethanol will dissociate that because it's going to break that disulfide bonds. So it's going to go from a cysteine to a cysteine. So we've talked about that. So you do add beta-mercaptoethanol in your sample, especially if you're trying to figure out if this protein, what's the molecular weight of this protein? Is it a dimer? Is it a monomer? Right? So you want to make sure you have that ingredient in there. Then with the proteins, the negative charge, this is what's important, the negative charge on the sulfate group is going to swap the charges on the proteins and make all the proteins in your pool negative. So it's going to be running based on charge, not molecular weight, but charge. Okay, so when you do run it, you make sure you hook it up properly so that all the proteins will run from top to bottom. If you hook it up on the wrong direction, it goes all the way up, right? You lose your sample. Okay, so it's good. The proteins are going to be running all these charged with negative, and they will move towards the positive charge. And I can tell you from experience, I've done it a couple of times. Okay, so that's normal. So with this SDS page, it's used to detect purity of the protein. Because obviously when you're done with the protein purification, you want to run it on a gel, you want to make sure it's the right size, but also if it's pure. So you want to detect one band, right? You don't want all the mess that comes with it. Then you know if all the mess comes with it, all those 10 bands come with it, then you know you haven't purified properly. And of course you need to know the molecular weight, that it's running at the molecular. Just because you have a band doesn't mean it's your protein. You better make sure that it's the right molecular weight. Okay, so for example, I'm just bringing you a stained example where you see different, I'm just using this as an example for purification purposes. You do not want to see this, right? If you're purifying a protein, you don't want to see this or this because there's so much impurity. You want to see one single band like this. Okay, so now when you look at this guy, this is also important for you guys going on to the lab, even though this is protein, you're going to be doing something like this for DNA. So what you do is, obviously, you're going to run your samples, whatever that sample is. And you're going to figure out what the molecular weight is. And sometimes what we just do is we run a marker, and a marker is a fixed molecular weight. So for this proteins, we've got myosin, which is 200,000 Dalton, all the way to a lysozyme, which is about 14,000. So that's your molecular weight. Okay, so bands will show up at that particular range. And on this sample here, that's your sample, you run it. So we can actually eyeball it and say, okay, maybe it's about 40,000, I've got my protein of interest. It's pure. But then what do you need to do? You need to do an assay. One thing you will do is not just eyeball it, but you want to get your accurate molecular weight of, say, your nucleic acid that you're going to run. So what you do is, on the y-axis, you take the logarithmic scale of all of these values, and x-axis, what you do is, you take a ruler, really a ruler, measure from the well to each one of these bands. And that's your standard curve. Okay? So once you have a straight line, all you have to do is, again, go back to your sample, measure from the well to your molecular weight, right? Take the inverse log, and you figure out what the accurate weight is. So standard curves are important because we want you to, number one, know how to use it and come up with an accurate value for the molecular weight of the protein. Okay, that's how we get the accurate weight of the molecular proteins using a standard curve rather than just eyeballing. Okay, so that's just something that you guys will be doing, and so you have a little bit of information background on this. Okay, so now we go back to more methods of isoelectric focusing. And so what isoelectric focusing does is it's depending on PI. Okay, so you've got a buffer range on the column that has different gradient, pH gradient. And you throw in your pool of protein, and your protein will migrate until it stops at the PI range, right? The pH at which that particular protein is at 0. That's your PI, that's the definition of PI. Okay, so start migrating, some proteins will go at 6.5 based on the PI, some will go at 5.5. So it's basically separating it out based on the isoelectric point of that particular protein. You guys are doing just one single protein, one single amino acid. But you can do this, like I said, for the entire protein sequence, whether it's 50 or 2,000 amino acids, all you gotta do is remove ionizable group and figure out where the 0 is and take the average. If you want a little bit more challenge, you can do that at the end of the chapter and see what the PI is. Okay, so that's basically what this model is of isoelectric focusing. You have a buffer with a gradient, and the proteins begin to migrate. So for this one, the orange one begins to migrate, and it stops here. That's where the band is. All right, and if you look at the pink one on this side, it moves, and at this particular pH is its isoelectric point, and there's your band. So each band will pop up at its own isoelectric point. Okay, so that's isoelectric focusing, all based on PI. You could do a two-dimensional gel electrophoresis where you separate based on PI and molecular weight. Okay, so that's two-dimensional. So on your horizontal, you're separating it based on PI. And then, remember, you can have a protein that has the same PI. So let's take an example with the pink one. And it starts to run based on molecular weight, right? And so in this particular case, this pool that has a particular PI actually is 4 different proteins. Okay, so that's how it runs. So it's based on PI, and then you separate it out. That's why it's called two-dimensional. You separate it out based on molecular weight. So this one here has 1, 2, 3, or 5 proteins in it. So it's these 5 proteins have the same PI, but different molecular weight. All right, so when is this useful? Well, this is useful when, first of all, let me show you this. So here's this isoelectric focusing. You see lots of little dots, and these are proteins that have been separated based on PI and molecular weight. When is this useful? Well, you can run a normal sample versus a tumor sample, and what you get to see is the intensity of the proteins. So in this particular case, what they're showing you is this arrow. And what you see this arrow pointing is in normal, this band doesn't show up dark, while in the tumor, it shows up dark. So what does that tell you? That this protein is subject for investigation, right? Why is it high in these tumor cells? So that's how we use two-dimensional electrophoresis. Do you guys have any questions so far? Okay, so let's look at what I came back from the beginning when I told you about specific activity. And specific activity, I told you, is total enzyme activity over total protein. So let's just look at all these numbers. Let's just keep an eye on specific activity and total protein. Let's not worry about the rest. Okay? So what you're trying to do is you're trying to figure out if you can purify the protein, and it has to be pure. That's the ultimate goal. So notice in this experiment, this person did 5 different types of steps to get a purified protein. So homogenization is really what we talked about first, breaking the cell open, where you get a lot of the proteins. So there's about 15,000 milligram of protein. And they did salt fractionation, ion exchange, molecular exclusion is another way for saying gel filtration, and affinity chromatography. So they had to go through all these steps to purify the protein. So the first thing you notice is we go from a 15,000 milligram of protein in that particular soup because you broke down the cell, so you got a lot of protein. And by the time you get to the end, yes, we have a small amount of protein, less than 2 milligrams, but it's pure. Once you test it out and see there's a single band and you do an assay, you've gotten the protein of interest. So this is about all we get. We don't get a lot of protein, but this lasts us for a while, depending on experimentation. So when you look at specific activity, you look at your total protein, your enzyme activity. Here's your enzyme activity. Total activity is here. So with just your protein of interest, it has about 52,500. You just take those numbers, between these two numbers, and you get your specific activity. So specific activity has to go up as you go through the number of steps, because you're purifying the protein in its purest form, but you're going to have less amount of protein. So that's why your number at the bottom is going to be smaller, which obviously is going to raise your specific activity. Okay? Any questions on that? Okay, so that's basically all these steps, and that's what's shown here. Okay, so you go through each of these steps from homogenate, breaking up in a cell, all the way to affinity chromatography, and you run it on a gel, and this is exactly what you want to see. You want to see a single band. Notice how impure this is, all the proteins. That's why you're seeing about 15,000 milligrams of protein. As you go from step 1 all the way to step 5, you should be reducing that number. And that's why you're starting to see the bands disappear or getting lighter as you purify it further. And that's what you want to see at the end. You want to see a purified protein. Okay, now I'm really going to go through, so I've done that. I'm going to go through the immunochemical assays. All right, so I already talked a little bit about gel electrophoresis, how that works, right? Swapping of all the proteins and making it all negative charge. And so it's going to run. And it's going to run towards the positive charge. So these are the ones, stars are the ones you're responsible for. Immunoprecipitation, Western blotting, and ELISA. Immunocytochemistry is also used with antibodies, but what you do with that is you use it mainly for tissues. All right, so that I'm not going to talk about. But all of these techniques use antibodies in some form or another. This is maybe a 15 seconds crash course in immunology, but that's all you need to know right now. Okay, so here's your antibody structure. I'll use this. And this is also a great example because it has disulfide bonds. So the antibody here has a heavy chain and a light chain, and they're linked together. These polypeptide chains are linked together by disulfide bonds. Right? So there's your disulfide bond created. How would you break it open to see? You would just take this and you throw in some beta mercaptan ethanol. It'll break these disulfide bonds. All right, so once you do have that, you notice that there is something called an antigen binding site, and that's where your antigen or your ligand is going to bind. So the antibody here will bind to the antigen. Okay, and that's what we're going to use, that principle where you have the antibody binding to the antigen. Okay, so here's your other model of interaction where you have the antibody and you have your antigen binding to the antibody. Everybody heard the term, right? Antigen? Okay, it's very specific for the antibody site. We have two types of antibodies, polyclonal, monoclonal. A polyclonal is not as specific, but it's cheaper to buy. Monoclonal is very specific. Okay, so it's a very pure form of antibodies, and of course it's going to be more expensive to buy. Antibodies take a long time to make. Thankfully, we have pharmaceutical companies readily available with antibodies that we can use for research. So the way that works is, it's interesting the way it works. So we use actually cancer cell myeloma, and we inject a mice with antigen, and you want to get antibodies out of that, and you want to grow a lot. So what we do is we take this and mix it in with the myeloma cells, and the myeloma cells will grow profusely, as long as it has a particular enzyme. I'm not going to tell you all the names of the enzyme, but as long as it has a particular enzyme, it'll grow profusely. Without it, it won't grow. So then it'll grow, you induce it, and you see these tumors are going to release a lot of antibodies, and you obviously have to sacrifice these mice and get the antibodies. Okay? And that's in a kind of nutshell. So some of these antibodies, like I said, are monoclonal. They come from a specific B cell. I'm not going to go through B cells and T cells and NK cells. That'll all come in immunology. So when you fuse it, you create a hybridoma, which is a mix, and then you get a clone of a particular antibody of interest. Or you can have a polyclonal from an anti-sera that has a mix. So it's a little less non-specific, but you will still be able to use it. Okay, so those are two types of antibodies. Okay, so let's go with the techniques now. Now that you know that the antibody binds to the antigen, let's look at immunoprecipitation. So immuno, as obviously when that name comes up, you know that some form of an antibody is used in the technique. So here in your test tube, you have lots of different proteins. Okay, and you throw in your antibody here. And of course you know what protein you're looking for, right? So you can actually purchase that antibody against that protein. So you throw in the protein, you throw in the antibody, and that antibody is supposed to recognize that protein of interest. Your protein is equivalent to your antigen. Okay? So here's the interaction between the antibody and the antigen. You also have beads thrown in it because you want to precipitate it out. Right? You want to plumb it down. You have beads thrown in it, which will complex to the tail end of this antibody. And then you spin it, and this will plumb it down. Okay, so you centrifuge it, it's heavier, so it'll come off on the bottom. And you just throw away the top and isolate it, take the bottom part of it to precipitate, and then we've got different detergents, which I'm not going to go into details, to separate out the interaction between the beads and the antibody and the antigen interaction. Okay, so the crust of it is you're fishing, right? You're taking the pool and you're trying to fish out your protein of interest. So it makes sense if you know what the protein you're looking for, and if this antibody is readily available, you could actually fish it out. Okay, I think this is the one that the book gives you. Here's your estrogen receptor that you're trying to look for in this pool. So based on that, you take your antibody-bound beads and you throw it in, and there it is fishing out the estrogen receptor. It comes down, you centrifuge it. Once you centrifuge it, like I said, you use a detergent, a denaturant, that will disrupt that non-covalent interaction that's occurring between the antigen and the antibody. And what you're left with is your estrogen receptor, because that's what you're fishing out, your estrogen receptor. Do you guys have any questions about that? How to do immunoprecipitation? Okay. ELISA is also another technique to determine if you have a particular antigen or antibody. Either way. Alright? And it's interesting that you can quantify the amount of protein. If you can figure out if it's a light-based spectrophotometer through absorbance, you can figure out how much of a protein you have. So the principle behind this is you have a plate that has an antibody. It could be a plate, or like I'll show you in the next couple of slides later, I'll show you that you can also use this on the gel electrophoresis gel. So you have a plate, you have an antibody that's attached to the plate, you throw your serum. If your serum has that protein of interest, it'll complex to the antibody. Then you throw in your secondary antibody, and that secondary antibody will complex to your protein. This secondary antibody has an enzyme conjugated to it. The reason it's conjugated to an enzyme is to figure out if you have that complexed to the antibody. So you throw in your substrate, and it usually lights up. Or if it's based on color, you can see the color difference, or the absorbance. Okay, so this is pretty much the same thing. So I'll go to the actual. All right, so this is an indirect ELISA. With the indirect ELISA and the sandwich ELISA, the indirect ELISA has an antigen bound to the plate. All right, so here's your plate, an antigen is bound to it. In the sandwich ELISA, I'm just going to show you the comparison. In the sandwich ELISA, your monoclonal antibody is bound to the plate. So with the indirect ELISA, you can test whether the patient's serum actually has the antibody present. This was a technique that I put out there for you guys. A company developed an ELISA plate that had the particular spike protein attached to the plate. Spike protein is the COVID. And then try to figure out if the patient has the COVID antibody. And I liked that one because it was actually in collaboration with the UAB. So I thought that might be interesting for you guys to look at. So here's the antigen bound. You throw in the antibody, and you can say that is your serum, right? If the serum has that antibody making it, then it will bind to the antigen. You wash it off, and you add your secondary. So the thing in black here is your secondary. So if the primary binds to the antigen, then the secondary will bind. So that's why the wash step is necessary. You wash everything off that hasn't bound. The secondary will bind. Remember I told you the enzyme that's conjugated? So it'll bind. So all of this is complex together. Then you wash again, remove anything that's not bound, and you throw in a substrate. Okay, I'm not going to tell you what the substrate is, but just say a substrate is thrown in. That enzyme will break down that substrate. Right, so this is basically what you've learned, right? E plus F mixed product. Right, so this enzyme will break down that substrate and make a product. In your case, the product is going to be the change in the color formation. Right, so you go from whatever color this is in blue to a yellow color. So that tells you that that particular patient has the antibody in his body or her body. Okay? So that's indirect. Same principle applies with monoclonal antibody. That's monoclonal antibody that's bound to the plate. So in this case, you're adding the antigen. And this may be useful for pregnancy testing. You know, you test the sera for a human chorionic HCG. So you test them for that. You throw in the serum, see if the patient has that HCG, and that particular antigen will bind. You wash it, throw in the secondary, and if the secondary will bind only if the primary is there. If there's a color change, that tells you it's positive. Right? So that's an example where you do have, you're testing for the presence of an antigen within the serum. Okay? And you can do that with HIV testing too. You test it for the antigen, the presence of an antigen, and that'll complex together with an antibody. Okay, do you guys have any questions about ELISA? Okay, so the last one technique here is Western blotting. So you're going to separate again using a gel, same principle that I talked about, and you're going to separate it out, and it's going to run through based on charge. And you'll be able to figure out the molecular weight. So we're going to use the same principle, and then we're going to throw in the antibody. So here's the gel running, okay? So all the proteins are here. Then you transfer it to a sheet, and all of these proteins will bind to the sheet. And then you throw in the antibody there. If that protein is here, is expressed, then throwing in that antibody should bind to that antigen or that protein, and it should light up. And that's what you see here. So you do the same principle like ELISA. You throw in the primary antibody, and then you wash it, do a secondary antibody, wash it, and then throw in the substrate and see if it's there or not. And that's a good method too, especially if you're comparing between two states, a normal versus a disease state, and you want to see if a particular protein is up or down. That's another way to look at it as well. So it's pretty simple, and this is an example of what a gel will actually look like. So here's your molecular weight markers, the standard markers, and here's your estrogen receptor, and you purified it, and hopefully these are different types of cancer cells. Here's a cancer cell, and you notice that they've stimulated they've stimulated with estrogen, and they notice that there's a high rise when you do that. All right, so that's just an example. So just by eyeballing, you say, okay, so estrogen receptor is about 70 kilodalton. All right, so do you guys have any questions? I want to actually have time to do the PI. No? Everything is good? Okay, so let's switch gears. Let's finish up. And don't forget, you have an assignment due today. It's now about 20 already in there, don't forget. The quiz on Thursday will be on this chapter. Okay, so the quiz on Thursday will be on what I did today. All right, so make sure you get that done. It's an online quiz, 5 questions. So these are the PI problems that I'm going to do. Pull that sheet out. All right, so we did alanine. Okay, so if I give you 2 pKa values, all you need to do is what? What do you need to do? Average it. Okay? But I'm going to go through it because I want to look at it in terms of the second question. Okay, so let's look at alanine. So that's alanine. Hydrophobic, how do I know that? Because it's a methyl group. 2. Alright, so this one, pKa that I gave you is 9.2. And carboxylic acid is going to be 2. So I'm going to remove the proton, the hydrogen, from smallest pKa value. And so what's the charge on this one? At a pH of 1? At a pH of 1, what's the charge on this? Positive 1. Good. Okay, so I'm going to remove the proton from the carboxylic acid. Okay, and so my charge is 0, obviously. And then I'll just take the other one, 2. minus 1. So far, so good? Okay, so that's pretty straightforward, right? So you just have to take the average, PI, you have two of them, you know that you just take the average, you don't even have to go to that step, unless you want to do it, because the second question, maybe you want to do it. And that's 5.6. Okay? So the next one is number 2. I ask you what the charge is at a pH of greater than 10. So at a pH of greater than 10, it's past the 9.1. So the charge will be all in this way. pH greater than 10, charge is negative. Right? Okay. When pH is greater than the PI, okay, this you can use when you have an amino acid with 3 ionized groups. When pH is greater than the PI, it'll be negative. So when pH is less than the PI, it'll be positive. When pH is equal to PI, it'll be neutral. You can use this, all right? You can use this principle. If you're given an amino acid with 3 ionizable group, and I throw in and say, what's the charge at a particular pH? You have to calculate your PI. And then you can use this principle for that particular pH. Everybody got it down? Can I take it off? Okay, so the next one I gave you was tyrosine. So tyrosine is a Y, right? One letter code is a Y. It's pretty much looks similar to phenylalanine, except it doesn't have that hydroxyl group, right? Phenylalanine won't have that hydroxyl group. So pKa, oh, I forgot to put the pKa's. Let me put the pKa's first for you. 2, 10.9, and 9.2. Okay, so you're going to take off the 1 here. So let me just go with what this charge here is. What's the charge on this? Plus 1. And negative 1. Right? Then we go to 9.2, which means I'm going to remove the amino group from the hydrogen from the amino group. Yes?
It will be.
Where? Oh, sorry, yeah. Goes from +1 to 0. Right, thank you. Okay, so we do cancel that out. So here's a plus, here's a negative, so it's a 0. So 9.2. So we're going to remove amino group, hydrogen from the amino group. Okay, so now we have -1. So at this point you can stop, right? And you can take the average, pKa values, 9 and 2. Let me just go one more step and complete it. Just 10.9. So we'll remove all the hydrogens now. So basic. That's my net negative 2 charge. Okay, so net negative 2. So PI, you know it's going to be, take the 0 value and take the 2 pKa's in between. Divided by 2 is 5.6. So anything above a 5.6 will be negative. The charge will be negative. If I give you a pH of, I don't know, 9.9, because it's going towards the negative. So anything above that range of 5.6 will be negative. Anything below a 5.6 will be positive based on what I told you, and anything equal to 5.6 will be neutral. Okay? Always use that, what I told you about pH, the relationship between pH and pI. Is that good? If this starts at a +1, look at histidine, look at arginine, that'll start off at a +2. Do an example with either histidine or arginine or both. And that'll actually start off with +2. What happens is, students will say, okay, I'm gonna start off with a +1, and I see this quite often. What they don't realize is, yes, you're gonna start out here, you're gonna take the first two, what happens with the +2 charge? Right, everything flows back one. Right, moves back one step. So make sure you do a histidine and an arginine. Okay. All right, so I'll stop here. Let me know if you have any questions for the review.